Good evening, everybody. Thank you for joining us. Apologies, we're just a little minute late opening up the webinar for you this evening. But I'm pleased that we're joined this evening by various industry experts and researchers that have been involved in the Forage for Change project that has been running and has recently come to a conclusion. I'm Katie James, I'm NSA Communications Manager. NSA has been very pleased to be involved within this project. And then joining us this evening, we have Dr. Mark Young from the UK Agri-Tech Centre, Adrian Thornton-Berry, whose sheep flock was involved in this project, Dr. Nicola Lamb from SRUC, and we're hoping we also have Ellie Roberts from NIAB joining us, although we think she may have been just slightly delayed, but no doubt she'll join us as soon as she can. This webinar is being recorded this evening, so if for any reason you want to watch back or you can't make all of the meeting, then it should be available on the NSA website at some time tomorrow. If you do have any questions, please pop them in the Q&A area of this webinar, and then we will bring those together probably at the end of everyone else's presentations. Okay, so I will pass over to Mark and speak to you later.
Thanks, Katie. I'm just going to share my screen now. I'll get Nicola or Adrian to give me a thumbs up. You can see that. Perfect. So my background is I'm a sheep geneticist by training and much of my work experience. But while working for the Agri-Tech Centre as a livestock specialist with ruminants as my feature, we've produced a number of reports about net zero, and I've got into learning about this methane business. And there's often talk about the fact that ruminants have always been here, and so we've always produced methane, so they can't really be the problem. But I've looked at the numbers, and there are some numbers which suggest that they are producing more methane than they used to be. So let's have a look at what that is. But we also have to think about the other context. That's why we have ruminants, and they're important for food security. So let's carry on and see what we can find out. Some of the data available online has provided estimates of the total biomass, which is the total weight of mammals that we've had historically and right up till now. And they measure this in terms of tons of carbon. And so there was 20 million tons of carbon tied up in mammals about 100,000 years ago when humans were hunter-gatherers. But by 10,000 years ago, when we'd started to move into agriculture, we'd knocked those mammals down to 15 million. And that big difference is what they call the megafauna extinction. It's when we were very successful at hunting them, and things like the mammoths and the woolly rhinoceros, they'd been good to eat, and so there weren't so many of them. But the point about that second box you see on the screen is humans are a thin blue line on the left. So the majority of mammals were still wild animals. So mammals have always been here, and due to human hunting, there was a reduction over about 90,000 years. If we jump forward to 1900, only about 120 years ago, we can see there's actually a massive increase in the weight of mammals on the planet. We've decreased the wild animals, the pale blue line. Humans have increased from just a thin blue line on the right to being 13 million tons of carbon. So the human population has dramatically increased between 10,000 years ago and the year 1900. So that's the success of farming, feeding people, and the huge expansion of the human population. We also suddenly see a whole lot of animals that are livestock. This is the brown bar. And effectively what that's saying is we've been very successful at farming, our population has expanded, and both on the basis of we were well fed because we had animals, our population has increased, so is that of the livestock to feed the human population. If we jump forward again to just very recently, some good figures from 2015, we see an even bigger increase in the weight of mammals on the planet. So what we've got now is even fewer wild animals. We've now got 3 times as many humans in terms of weight of carbon as we had all mammals back 100,000 years ago. And the livestock are even bigger again. And this reflects the fact that human populations continue to grow. We all know about human population expansion, and livestock have grown with it in order to feed us. And this is because agriculture has been so successful. As we've increased our footprint on the planet, we've seen more humans and more animals being used to feed us. So mammals have always been here, just not that many. That total in the bottom line is 170 million. So that's about 8 times more than we used to have. And so what the question is, how many of those are mammals? If you look at this next graph, looking at it just now, about half the mammals are ruminants, and these are the ones that produce methane through the fermentation in the chambers in front of the true stomach, what we call the rumen. And so if we've got 8 times more mammals and half the mammals are ruminants, that's 4 times as many as we had total mammals before. So we must be producing more methane than we did historically, simply because we've got more mammals, and that comes from agriculture, and the ruminant animal has been a very successful animal in agriculture in terms of producing milk and meat. Now, the key thing about these figures in terms of livestock is it doesn't include poultry. But what we're seeing here is a massive increase in the number of mammals, of which a high proportion are ruminants, and so we produce more methane than we did many, many years ago. And so effectively, the global warming that's occurred has been partly caused by the increase in the number of mammals, of which a high proportion are ruminants that we farm. At the same time, in order to farm them, we've been clearing forests. The best estimate I can find from looking at the literature on the internet is we've lost about 50% of our tree cover, and that's because we've cut down trees in order to grow pasture to feed animals like ruminants, as well as clear some land for arable farming. And that loss of carbon from the trees has been affected the amount of carbon given off, but it's also meant that we don't capture quite as much carbon or quite as much carbon as fast as we used to. The challenge here, and I think the science is still developing, is what proportion of carbon is contained in a hectare of forest versus a hectare of grassland. And more is kept in grassland than appears from the surface, but I don't know quite how we equate them as yet to quantify it. So the reason all of this has occurred, of course, is because of that massive increase in humans. We've got past the peak rate of growth, but the population numbers are still increasing. And so those big increases since 1900 in the number of livestock and the number of humans is just again shown on this graph. And effectively, I would say here, the genie's out of the bottle. You're not going to be putting reducing the need for ruminant livestock unless you reduce the need for human food. And so I think that genie is well and truly escaped, and we need to be thinking about how we farm animals as opposed to we must be getting rid of animals. So let's look at what the conclusion is. Simply, ruminants have produced methane always, and it's part of a short carbon cycle, which we know is quite different to the long carbon cycle, where we burn carbon that's been hidden underground as coal and oil for millions of years. So the short carbon cycle is good. We've just got many more ruminants than we did historically. And most of the growth in livestock numbers and ruminants has occurred in that last 100 years as the population growth has taken off. So what's the story with the need? Ruminants have contributed to global warming, and we really need to make sure we reduce the methane intensity for the amount of food we produce. But important thing is to think about where ruminants fit into our food system. And some very elegant work done by the agricultural scientists in the Netherlands at the Wageningen Research Institute have said what we should be doing or considering is having animals not competing for land with human food crops. So arable land should be devoted to growing food that humans can eat, and animals should be eating either byproducts of the food system or eating things that humans can't digest and get effectively much nutrition out of, or be fed forages grown on land where we can't grow human food crops. And if you do this, you get the most efficient farm system. And so if you don't have animals in the system at all, their research showed that we get a total nutrient production is reduced, we reduce the quality of human diets, and it's less efficient use of our agricultural resources. And the key agricultural resource is land. Another paper by the same research group has come up with an elegant idea called the land use ratio. And the simple question is, can you produce more plant protein or animal protein on a hectare of land of a particular type in 1 year? And so if you look at the two types of land depicted here, effectively we're talking about how suitable the land is for plant food production versus animal feed production that production then turn into animal food. So basically, from the previous slide, we'd say animals should primarily be fed food production byproducts and the lower quality foods that humans don't eat, and forages grown on land that you can't be used to grow plants. And that's effectively what we've got here. The sheep is an ideal animal for these harder environments where it's not suitable for growing human food as plants. And that is one of the key settings for this particular project that we're talking about. So effectively what we're saying here, the key role for ruminants in our food system is to produce nutrient-dense foods from land not suited to food production from plants, but we should be looking at efficient ways to do that to minimize the amount of methane we produce at the same time. So that's it for me, Katie, and I will now switch off once I figure out how to get rid of that bar there. Stop sharing. And it's ready for Adrian to give his outline of the project.
Great. Thank you very much, Mark. That's really good scene setting, and there's a lot of challenging things in there. But I think as livestock farmers, I think what Mark's saying is a very helpful and balanced view. We're not the enemy, we are part of the solution. And in that vein, I am to try and explain my background. I was born and raised here in Wensleydale. We've got some better land that you can run a tractor over and mow, but I would say nearly 2/3 of our land is either on the side of a hill, very bumpy, gravelly land. Down in the bottom, about 500 foot is our lowest point, and then we rise up to 1,850 feet at the highest point, and we have some enclosed moorland up there. Everyone knows as allotments, which were all pretty much hammered in the 70s and 80s when we were all pushing as many sheep out as we possibly could. That sort of landscape now we've had in stewardship for a bit, and that's recovering. But we've had a relatively easy ride, I would say, or helped cushioned along by the good old area payments for the last, it's nearly straight 20 years, isn't it? 2005 to 2025. But that crutch is gone. Even with the new stewardship schemes, it's unlikely we're going to be able to recover the business premium scheme payments. So myself and others in the locality have been sort of focusing our minds on how government's encouraging us to try and get private finance to basically replace the government support for our physically and socially challenged farming areas. So hill farming is, I think, in a real mess, and there's a lot of people very worried. So why this project? We've all been racking our brains to think of how we could promote what we do and help support the farming community, and looking about what we do. And there's a definite economic driver for sheep production to get onto a more lowland type commercial breed with a bit of housing, a bit of extra feed, and increase the lambing rates, weaning weights and everything, generally a more marketable lamb rather than a smaller hill breed for sale. I'm not including the breeding market, obviously, the mule production. But So the drive is to be more commercial, but is that the best thing for society as a whole? What can we give? What can the Swaledale sheep, which is one of our foremost native breeds, it's actually the badge on the Yorkshire Dales National Park. I mean, it's really iconic breed. Surely it, who's in the area that it was raised and genetically designed for, will be a more useful and adaptive breed for the landscape that we have and the sensitive habitats we have, than the more commercial breed that's designed to live on rather better going. And that was the trying to find this sort of silver bullet, if you like, a message we could sell that our sheep, these Swaledale sheep, are better for the environment and society as a whole, rather than the commercial breed on these sensitive habitats. So that brought us into a straight comparison between Texels, cross lowland breed, and our Swaledale, pure Swaledales. And the design of the experiment, which will go into Nicola from SRUC will talk about. Basically, we had to sort out 60 ewes of Swaledale and 60 Texel ewes, and we had to get all the data for each ewe, and Nicola will go through that, but it was quite a complicated task, and we had to individually record each sheep before we split them up into the batches. So there were 20 of each breed type on each forage type. So we had 3 groups of 40, 1 group on improved pasture, improved grass on which fertilised sewage, slurry on, which is ryegrass pretty much and not much else. And then we've got these long-term stewardship areas, which are basically very herb-rich, lays on very poor, very free-draining moraines in the bottom, which you can't really mow, and that hasn't had any fertiliser or chemicals on for over 30 years, and it's very floristically diverse. So we put 1 lot on there, 1 lot on the improved grass, and then another lot on this regenerating moorland, which was mainly bilberry, that heath straw, and generally moorland plants, but up at sort of 16 to 1800 feet. So we put all the breeds out up there, and then we had the joy of up there for 2-3 weeks, and then gathering them, bringing them down and testing them. It was quite a intense few days with all the measuring, and the sheep did lose condition. I personally think probably because they were chased about quite a bit, gathering them. We had to keep gathering, bringing them back and shoving them backwards and forwards. But anyway, Nicola can comment on that. But although it was very stressful and it's taken a lot of time, but we've, I think, put together an experiment which has shown there is a benefit from swales. Nicola will go into the results in more detail, but definitely we might be onto something here. But there are other variables we're going to have to consider if this work's going to be taken on further forwards, and there is a appetite to support traditional breeds in this traditional landscape. But we have to find a mechanism to get money from the corporate sector to farming, and I hope this experiment will facilitate this. But we've got to pick up the baton and do a bit more work. But it's we're on to something, and you'll hear more later from the NIAB lady Ellie explain why sheep on moorland might be a good thing for the society as well. Anyway, I think that's probably enough from me, Katie. How's that sound? Is that long enough for you?
Yeah, absolutely fine. Thank you, Adrian. So we'll now hand over to Nicola, who's going to tell us a little bit more about some of these results from the study.
Okay, thank you. Hopefully you can all see that in full screen now. So Adrian and Ellie Rayner from the Game and Wildlife Conservation Trust had kind of thought up this project to try and answer some of those questions that Adrian spoke about. And they came to us because at SRUC we have the capability of measuring methane emissions from individual sheep. And this was one part of the puzzle that Adrian wanted to look into in a bit more detail to see what the methane emissions were likely to be from different breed types that were grazing the different forage types on his farm. So it wasn't really an experimental design that we'd used our equipment with before. So it took a bit of thinking about and a bit of planning to see if we could try and get a handle on how the different upland breed types and the different forage types were affecting methane emissions from the sheep. So we'd done a bit of research at SRUC, and some other groups around the world had done similar research, looking at methane emissions and factors that affect methane emissions. And the work that we'd done with UK hill and upland sheep to date really suggested that the types of sheep that we keep in the hills and uplands tend to have larger rumens than the breeds that are more generally in the lowland type systems or the terminal sire breeds relative to their body size. And these larger rumens have probably evolved to help these animals digest the poor quality forage on the hill type pastures. But there's been some evidence to show that larger rumens can lead to greater methane production. And we know from selection lines of sheep in New Zealand and Australia where they've selected for high or low methane emissions, those of the higher methane emissions also have larger rumens. So there's some evidence that these hill breeds also produce more methane than some of the lowland breeds or crossbreeds relative to their body size. But most of these trials that had been done before had been on research farms and generally compared lambs that were all being grazed on low ground pastures or in indoor feeding trials where animals were much more easy to closely monitor. But within this project that Adrian and Ellie had designed, what we really wanted to look at was emissions from ewes grazing typical pastures that we find on upland farms in the UK. So we designed an experiment that Adrian mentioned there. We used mature ewes and we had 120 ewes in total that were on the trial. So it was just in one summer, autumn season, and we used the ewes in mixed breed groups on the 3 different vegetation types that Adrian discussed before that he has on his farm. So the improved grassland, the species-rich grassland and the moorland. So they were our 3 forage treatments or forage types. And then we had our 2 breed types. So we had the pure Swaledales and we had the Texel crosswale or the crossbred animals. So at SRUC we have our portable accumulation chambers. So the trailer that you see at the top there has 12 boxes on it. So they're aluminium boxes about 1 metre cubed with a perspex window at the front. And we can put animals in them for a short period and get measurements of methane emissions. So the plan was to take the trailer down to Adrian's farm and measure them twice, so repeated measurements 2 weeks apart to measure the methane emissions from each individual sheep. So after the lambs had been weaned from the ewes went out onto our 3 forage types. And when we say forage types, it was 1 field of each of the 3 different types. So there were 40 ewes on 1 area of moorland, 40 ewes on 1 area of species-rich grassland, and another 40 on the improved grassland. So they went out onto these forages for about 3 weeks, and then they were gathered in and went through the pack chambers down at the steading, and then they went back out onto the same forage treatment and were gathered again 2 weeks later and had a second pack measurement taken. So we tried to well, when I say we, Adrian and his team tried to gather the sheep off the grazing areas at the same time, so that they'd been off feed about the same length of time before they went into the pack chambers, because we know that time off feed can affect the methane readings. So they were gathered from the 3 different areas. They were weighed and allocated to their batch of 12 to go through the pack chambers at the same time. And we tried to balance each of these groups of 12 for breed and forage type. So as I said, we've got the 12 chambers on the trailer to measure emissions from individual sheep. So one sheep goes into a chamber at a time. They've been off feed for a minimum of 1 hour before they go into the chamber. And then it's just really an empty box and it's sealed, so we can measure the gases from inside the chamber. They go into the chamber for 50 minutes and we measure the gas in the chamber at the beginning, at 25 minutes and at 50 minutes, and then the animals are released. So what we're measuring is changes in methane, oxygen and carbon dioxide. So the methane and the carbon dioxide go up, and as the oxygen is used up, the concentration of oxygen comes down. And this has been developed in New Zealand and Australia, this system, and they've done a lot of developmental research to show that this 50-minute measurement accurately predicts the methane and carbon dioxide emissions per day from the sheep. So we then extrapolate up to grams per day when we're giving our results. So another measurement we took were live weights of the animals. So we weighed the animals at the start of the trial before they went out onto the different pastures, and we took a body condition score at that same point. And then the animals were weighed before they went into the pack chambers for the first and second time. So there's quite a lot of numbers on this table. Sorry, the top ones are the Swaledales, and the bottom ones below the middle line are the crossbred animals. So you can see that the Swaledale ewes were lighter with a lower body condition score at the start of the trial. And so that body condition score is measured on a scale from 0 to 5, and the swales were about 1.3 higher in terms of the condition score than the swales on average. Sorry, crossbreds were higher than the swales on average. As we went through the trial, the ewes lost weight, and the crossbred ewes lost a higher proportion of their starting body weight than the Swaledales. So the swales lost on average 2.7% of their body weight, and the crossbreds lost 4.9%. And this was mainly before the first pack measurement. They then put on a bit of weight in general between the 2 pack measurements across most of the groups. And when we compare the different forage treatments, it was the ewes that were grazing on the moor that lost the most weight. So the Swaledales that were on the moor lost about 4.5% of their body weight over the 5-week trial, and the crossbreds lost 7.6% on average. So that may be a reflection of the feed availability or the quality of the feed that they were getting in the different areas. So looking at the emissions data that we got from our pack chambers, you can see a picture at the top there of Karen, the technician, taking some of the gas measurements, and you can get a better idea of how it looks inside the trailer there and the size of the chambers. So we're measuring changes in methane and carbon dioxide while they're inside the chambers, and then we can extrapolate this up to grams per day. So the methane tells us about the digestive process in the animals, and this is controlled by the bugs or the microorganisms within the rumen. And you can see here in the blue graph, the box in the middle shows where 50% of the data are, and then the whiskers either side show the range of data that we saw, with some dots outside those whiskers being outlying values. The crosses in the middle of the boxes are the average values, or the mean. So the crossbred had a slightly higher average, and a wider range than we saw from the Swaledale when we just looked at the measured methane emissions that were calculated. When we look at the carbon dioxide and the green graph there, again, the crossbreds produced higher levels than the Swaledales and had a higher range in values. But these are the what we would call the raw methane measurements, and we were a bit unsure how they were affected by other things, other sources of variation in that data. So although they were all mature ewes of a mix of ages, there was a slightly different age profile for the two breeds. So there were a few more younger ewes in the crossbred group than there were in the Swaledales. So we wanted to kind of make sure that any changes we saw weren't because of differences in age profile. We also know there's differences due to the measurement round, so whether they were, when they were being measured after 3 weeks or after 5 weeks, the vegetation would have been different that they were grazing on just as the season progressed. And also there may be changes due to the batch that they were in. So that's a batch of 12 that they were measured in. So as the batches progressed through the day, they would have been off food for longer. So we took 3 batches off the fields and measured 3 batches before we collected or gathered in the next batches. So there would have been effective measurement batch as well as measurement round. And there's also this difference in live weight as well. So any differences that we see with the crossbreds producing more gas, is that just because they're bigger animals? So we did some statistical analysis to adjust for all these different sources of variation to try and get a fairer look at the comparison of the different breeds and forage types after we'd adjusted for changes due to these other sources of variation. So once we'd accounted for these other effects that we knew caused variation in methane emissions, we didn't actually see any significant breed difference. So once we adjusted for differences in live weight, the swales and the crossbreds were producing around about the same amount of methane. We also didn't see any breed by forage type interaction, which means that the forages were ranking similarly between the breeds. But what we did see when we ran these models was that there was a significant effect of forage type. So the ewes that were on the improved grassland were producing more methane than those on the other 2 forage types, so the species-rich grassland or the moor. And the difference between those 2, the species-rich and the moor, wasn't actually statistically significant. So we couldn't have confidence in saying that they were any different from each other, but they were both lower than the improved grassland. In terms of the carbon dioxide emissions, there was a significant breed difference even after adjusting for live weight and other effects in the model. So the crossbred ewes were producing more carbon dioxide than the pure swales. Again, there was no forage by breed interaction, so we saw the same rankings across the different breeds. And we saw a significant effect of forage type again. So again, the improved grassland produced significant, the ewes that were grazing it produced significantly more carbon dioxide than the ewes on the moor. But this time, those that were in the species-rich grassland produced as much carbon dioxide as those on the improved grassland, which was more than those on the moor. So just to conclude what we saw from our methane measurements, in terms of the breed effects, the methane and carbon dioxide emissions, we saw they varied within breed and across the breeds. And although the crossbred ewes produced more methane and carbon dioxide than the purebred swales before we adjusted for live weight, after we'd adjusted for live weight, there was no difference between the breeds. So that difference we were seeing was just due to the fact that the Texel crosses were bigger. And then after adjusting for live weight, we found that the Swaledale ewes still produce less CO2 than the crossbred ewes. In terms of the forage effects, and I'll just summarise these after we've adjusted for live weight. The ewes produced more methane and carbon dioxide when they were on the improved grassland compared to the ewes on the moor. And the ewes produced similar levels of methane on the species-rich grassland as on the moor, but produced similar levels of carbon dioxide on the species-rich grassland as on the improved grassland. So one thing that we couldn't tell from this trial is whether the differences between the forage types were because the animals on the moor were eating less. There was some indication that this may be the case because they lost most weight, the groups that were on the moor. But it would be really nice to know if these differences were due to the amount that the animals were eating, or whether it was all due to the different species types that they were eating. So our take-home messages from the methane work were that we hadn't seen any evidence that the Swaledale sheep were producing more methane than the half-bred ewes when they were grazing the different upland forage types. And this was despite those earlier studies where we'd compared the hill type breeds to lowland type breeds and found that they had bigger rumens and produced more. In this study, we didn't see a difference between the breeds, with the swales producing any more methane than the crossbreds. One thing that Adrian hinted at was that this is one part of the puzzle, one part of the information that we can consider when we're looking at the impact of the different breeds and upland system types. But it'd be also really nice to have a much larger project, or to look across a number of farms and consider the data on methane alongside data on production, ewe and lamb survival, days to slaughter of the slaughter lambs, product quality, inputs, etc. But there is evidence that grazing different upland forage types at different times of year may affect the emissions that we're getting from these ewes, potentially linked to the intake of available forage, but it'd be good to expand our research to see if we can answer some of these other questions. So that's all I wanted to say on the sheep side of things. Thanks very much, and I'll hand back over.
Thank you, Nicola. So if we've got Ellie, if you're able to join us now. I think you're here.
Hello.
Hi, Ellie. There we go. Hi, Ellie. Have you got your slides to share? I did see some came through to me, but I
have. Yeah,
brilliant. Thanks. Over to you then.
There we go. Have they come through okay? Lovely. Okay, let me see if I just check I can get the... There we go. Okay. I'm the forage crop specialist at NIAB. My role includes a few different types of job. It's looking after the recommended lists for grass and clovers, as well as other forage crops, and then various technical inputs in various ways. So this has been a really interesting project to chip into a bit. My role has been to have a look at the data that's come in from the assessment of the 3 different grazing forage types, and see if we can draw any conclusions from that. Now, unfortunately, project wasn't big enough to be able to get data about how much the ewes were consuming of each of the different plant types. So we can only make assumptions based on what we expect them to be doing in a bid to hopefully get further research happening down the line. So of the 3 forage or species composition types, we had, as Adrian was explaining earlier, and Nicola, we had the heathland, hill, rough grassland. So this is more of your typical upland grazing areas that tend to be thinner soils, but not always. Usually, of course, harder to get up and do any cultivation. So there's not a lot of choice for what grows there, and not a lot of option for increasing or changing the species present. And certainly restrictions these days on the amount of nitrogen or anything you can do to help improve those pastures. So one of the main major species was the bilberry, as Adrian said, covering around about a third, and then red or sheep fescues also. Now these species are generally quite, they're not the most rich species. They don't have really high sugars throughout the season, but there is a lot of forage to digest, and those sorts of forages, if they're rougher forages, a lot more cellulose, a lot more lignin, can lead to the production, but don't necessarily in this case, they can lead to the more production of methane in the rumen. But then other plants such as the bilberry and heather that may have been present in some areas as well, certainly over here on the North York Moors where I am, they have certain secondary metabolites inside the plants that interacts with to a certain extent with the microbes in the rumen, and they can capture some of those free hydrogens that float around when digestion happens, and then those hydrogens can't join with the carbon dioxide and create methane. So the presence of some of these species in the rumen can help to reduce the production of methane, even though some of the rougher species themselves could increase the proportion of methane production. So it's kind of cancelling the other out. In the species-rich grassland, it all looks almost like a very neat kind of strange golf course setting from here in this particular picture. It's kindly given by Ellie Rayner, who was leading on the project, and I don't think is with us today. So yeah, again, more of the fescues. So a really good hardy species of grass, and fescues are actually crossbred now with ryegrass species to create festiloliums that you may well have heard of or used yourselves. And the reason for having the fescues in is because they're usually good rooting. They're able to make use of perhaps poorer soils, and they tend to cope better in less productive environments or drought conditions, for example. So they're becoming much more popular in recent seasons. So again, through the mixture of species there, you'll have some that are more and some that are less digestible. On the whole, salad burnet as a herb can be helping potentially to counteract some of the methane production, but I don't have any enough data on that to be conclusive. And yarrow as well. As well, white clover isn't a big factor in the balance of methane, but important, of course, for getting a good bit of protein into the animals. And then the improved grassland was predominantly perennial ryegrass. So not a lot of conclusions to draw from those really, other than the assumptions as I've just noted. Regarding the factors that affect the amount of methane that is produced and therefore emitted, it is a combination of that botanical composition, as well as the age and growth stage of the plants that are being grazed. So as Nicola was mentioning, the forage intake and the nutrient levels of those species can vary throughout the season. So clovers, for example, tend to be at their peak just as they're starting to flower. Whereas grasses, as they're starting to flower, they're really reducing their digestibility. And as not a huge amount of data available on different herbs and moorland plants that could be available. So the structure of the sward as well is important in the physical palatability of how easily animals can eat and get at it, and how the more older advanced plants, just like in permanent pasture, tends to become less palatable, and sheep will favour the younger leafier growth. The fermentation processes in the rumen is the major factor for how methane is produced. As I say, it's the free hydrogens that get released from the normal digestive processes of the bacteria and the other microbes within the rumen. And it's having something to mop up those free hydrogens as they're released will then stop them being able to combine them and make the methane. Then moving on from the animal and whatever it's belching out, we also have what is particularly interesting, I think, in this case, is the soil. Where the soil is well aerated, which won't be all parts of these moorland pastures, but certainly some areas where you've got good aerated, healthy soils with good biological activity beavering away, the presence of these grazing animals will stimulate the development or the further growth of these methanotrophs, so these bacteria that like to feed off methane, and they will capture and feed off the methane that is breathed out or belched out, sorry, from these users they're grazing. And in that respect, well aerated areas of moorland pasture do act as methane sinks, so they're helping to take out some of that methane from the atmosphere that's being released. And in that respect, good moorland habitats, as well as grassland habitats elsewhere that have got good soil structure, and particularly seems to be those that are related to woodland or scrubland in close proximity, will be really good for capturing that methane, bringing it down, digesting it into the ground, so taking it out of that cycle of emission release in the atmosphere. It also, with the heather and bilberry, as I say earlier, those secondary metabolites, polyphenols, condensed tannins, they help to bind and inhibit that production of methane in the rumen, and it will shift the types of volatile fatty acids that are produced, and therefore whether in how the animal is making use of those in its metabolism. Benefits of having diverse botanical swards is the legumes, of course, are capturing nitrogen, so helping to feed the rest of the plants in the system. Also, by having some of those legumes within the digestive tract, the better or higher amount of protein also gets a better balance potentially of energy and protein for the rumen microbes themselves, and also can influence the speed of rumen passage, and in that way help to reduce methanogenesis within the rumen. I've mentioned some of these things already, but there has been some work done about methane in these sorts of diverse swards. You get a lot lower, so 25 to 30% in some trials, lower methane production compared to grass only. Now, what we also have to think about is it's the whole system. So the more, it's very rare, or it certainly isn't around here, that moorland grazing is the sole grazing source. There will be some more lowland permanent pasture, and perhaps these days in recent years with the funding we've had through SFI, some herbal lays. Now these may be really diverse, purposefully sown herbal lays, or they may be diverse species grasslands, naturally like nice, more traditional hay meadow type settings. And in these, we're also then finding the herbs that contain these polyphenols that can help to inhibit methanogenesis. So the chicory and plantain that we get in them certainly tends to be sown. What we do get ribwort plantain naturally occurring in the UK, but the chicory and plantain varieties that are the most productive ones that are sown these days are ones that have been developed mostly in New Zealand. And their activity can help to reduce that methane production, as well as the efficiency of nitrogen metabolism in the animal and less losses of nitrogen, such as this 34% losses through urine in particular. So lots of ticking boxes for healthy environmental benefits of having these more diverse species within the system. So that combination of the moorland activity, your healthy soil for the diverse species in the lowland areas, and we're all around the system is helping to reduce the amount of methane that's produced. Yeah, also with more diverse species, we tend to get increased forage intake, higher palatability, more efficient feed conversion as well, and really good animal performance. Yeah, I think I've covered all of that. In the more species-rich upland and permanent pasture, so not quite your full herbal lays, but still some nice diverse mixtures. Again, you tend to have those dominant native grasses, which are slower to break down and therefore produce more methane in the rumen. But then we're having that mitigation with the legumes and the tannin-rich species with the secondary metabolites, the root polyphenols, reducing that methane. So in the whole system, we're seeing a really nice balance of what then comes out from that rumen. There isn't very much research data, but what is there suggests that the ericaceous species, so those heather and bilberry in particular, help to suppress that methane production of around about 40 to 50% when a 30% heather proportion was included. Of course, we can't assume that happened. Heather certainly wasn't a prominent species in the swards that we were looking at, but it's all worth considering. I don't need to do that again. Yeah, I've mentioned about the methane sinks and the root systems as well, releasing oxygen into the rhizosphere, further enhancing conditions to keep those methanotrophs happy, as well as capturing some carbon. So what I think also we need to bear in mind is the Swaledales as a breed are not in this setting are not only helping to maintain these habitats as methane sinks, but they're also, if they're grazing a wide range of these species, can be really efficient at not producing much methane for their productivity, be that lambs mainly or some meat. But also something like around an estimate of around 230,000 North Country mules are produced, mostly from Swaledale mothers. Massive contribution to the wider stratification of the UK sheep industry and the role of the Swaledale breed within that, as well as helping to maintain these native, I say native, natural. I think heathlands, if left to their own devices, would eventually turn into scrub and woodland. But by maintaining the habitats as they do, allows for more diversity to be present and to be looked after. So yeah, big role in the wider system to take into account. It would be great to have more data. Yeah, that's everything from me. Sorry if I've gone over a bit too long. Get a bit excited.
No problem at all, Ellie. Thank you.
Oh,
okay.
Oh, there we go.
Great. Thank you to all our speakers. So if anyone does have any questions at all, please pop them in the chat or the Q&A. Now we've got a little bit of time that we can have a bit of further discussion if wanted. Don't think we have any currently in there.
While we're waiting for the first question, Katie, I think the point is worth emphasising that Nicola made, that the adaptability and the suitedness of native breeds to native farming environments that Adrian's aiming at, it's more than methane. So it could be to do with the level of productivity, their ability to survive and maintain body condition and be up and ready for lambing again next year, that other breeds can't handle in the same conditions. So we need to really characterise some of that local adaptability by looking at a range of traits. But you know, any questions from the audience would be worth
Yeah,
worth us
so
addressing.
we have had one come in then, Mark. So I guess this is perhaps, well, I don't know, any of you may be able to answer this. So were the forages tested in vitro?
As I understand it, it was just assessment of the presence of the species. There wasn't any laboratory testing of their nutritional value.
I dug up the swards and took them to the Yorkshire show, but we didn't actually then show demonstration of the different sward types and a chap there, but we didn't actually cut any sections of forage and put it through I don't know, do you burn them up and see what's in them?
Yes, wet chemistry or near infrared. Yeah.
Basically,
Okay.
lab analysis.
Another question then is asking, did all the ewes from the trial have the same number of lambs? I suppose thinking that, you know, Swaledales will often just have a single, whereas these Texel crosses might be more likely to be carrying twins or more.
Yeah, this is back to the kilos of meat produced per available forage, I think, is what was headed at, and that's one of the variables that we were going to try and look at in the next stage, Mark, wasn't it? So we're basing mine, we're doing this testing in the autumn after they've been weaned. We didn't actually record what the individual ewes had prior to the testing per se.
They're about 30 or 40% lighter too, weren't they, in live weight? So in terms of weight of lamb per weight of ewe, the advantage of, yes, the Swaledales may have fewer lambs, but they're also got lower maintenance costs.
Yeah,
Nicola might be able to comment more.
I think ideally we'd have had ewes that were balanced for number of lambs produced, but with the largest numbers we used, we didn't have the information what each individual ewe produced, because they hadn't been recorded through lambing time and weaning. We just really recorded them from after weaning onwards, but as well we didn't have the balanced sample sizes that we need for that sort of
what were the average live weight differences, Nicola?
So I'll just click back to the swales at the start of the trial. The swales were 58.3 kilos and the crossbreds were 77.3.
Okay. Right, any further questions? No?
If anyone gets a chance to get involved with the pack changing bits, they are amazing bits of kit. I was very impressed by the design. It's New Zealand, is it, Nicola, that designed them, and it is very impressive.
I mean, there's 10 years worth of development that went into it. The first versions were nowhere near as sophisticated. I just did a quick calculation on my phone. The crossbreds were 33% heavier, so you'd want to see more than 33% extra lamb per ewe in order to make up for that sort of difference.
Question in the Q&A.
Yes, yeah, we have got a couple more just come in there. So we've got a question asking, if heather should reduce methane, why did the pack chambers not show much difference in the moorland grazing, or wasn't there much heather on the moorland?
So
there wasn't much heather cover. There was just bilberry, really.
So the one significant difference we did see was between the moor and the improved grassland. So with the moor grazing ewes produced less methane.
But the thought is that the other pasture, which also produced less methane than the improved, may have had similar compounds in the species-rich pasture, may it?
but also because we weren't able to measure intakes, it's difficult to know how much was being consumed, and that's going to vary throughout the season as well, through the different growth stages of the plants and the different nutritional requirements of the animals. So we need a lot more data really to be able to draw more useful conclusions. And
okay, and perhaps this might be our final question. So just thinking about sort of future studies, it says, will this experiment be repeated to account for different bioactive levels in the forages due to seasonality?
that would be wonderful if there was funding available.
Yeah, I think
Yeah.
the reason that we did it at that time of year as well was because it was easier to measure the ewes when they didn't have lambs with them. One of our trials at SRUC in our Hill and Mountain Research Centre, we're doing a similar thing with Scottish Blackface and crossbred ewes. We're measuring them in the summer and in the autumn. And yeah, it's quite challenging when you've got lambs to try and work with as well, and get the ewes in and out the pack chambers without mismothering.
I'd like to just stress the idea that the sheep are suited to this type of country. They produce a really high quality product under challenging conditions, and we mustn't lose sight of that. And we must avoid any unfair comparisons with the amount of methane or emissions that come from some plant crops that you just can't grow here. So they're doing a really fantastic job, sheep, at turning what is very challenging farming environment and turning low quality forage, low quality for human nutrition, into a really high quality food product for human nutrition.
And it's also worth noting that the crossbreds were crossed with Swaledale, crossed with something else, so it's difficult to know what genetics they were pulling over from their Swaledale parent, rather than if it was just a terminal sire breed on its own. That would be more interesting things to find out.
Okay, we have had just one more question come in before we finish. So have any microbiome analysis been done to assess any changes in the rumen?
So not in this project, but in the Breed for Change project, which is a larger project using similar pack chambers on across a number of different maternal breeds. Within that project, we're looking at rumen sampling some of the animals that we're recording quite closely for things like feed intake and methane measurements, and we're also CT scanning them to look at the size of the rumen and the body composition. So there's some preliminary results coming out of that, and there's definitely relationships between the different types of microbes in the rumen and the amounts of methane that are being produced, but we've not got the full picture from that. We've only analysed the first year of that data so far, but yeah, there's really promising results on that coming out of New Zealand and other countries as well.
Okay, and yeah, I think the Breed for Change project comes to a conclusion, doesn't it, in the next few months? So I'm sure there's going to be a lot more results and further webinars to share all the results of those.
Yeah.
It'd be surprising if the microbiome isn't involved, because I think that's what those compounds that Ellie was describing, they impact on the success of different microbes living in the rumen and how well they digest food. So it'll definitely have effects on the microbiome. The question is, does the Swaledale have a naturally different microbiome that gives it an advantage? So those are real interesting questions out there.
Okay, well, I think that sort of brings us to an end this evening. Thank you everybody for joining, and obviously a special thanks to Mark, Nicola, Adrian and Ellie for joining us and sharing your interesting information this evening. as I said, this should be up on the NSA website sometime tomorrow. So if you do want to share the information with any colleagues or friends or neighbours, then please do encourage them to have a little look when it's online. But otherwise, wishing you all pleasant rest of your evening, and thanks for joining us.
Thank you. Goodbye.
Bye.